Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to the 10th webinar in the Neurophysiology 2023 series. Today's webinar is titled Cellular Brain Repair for Parkinson's Disease: Is the Answer in the Biomaterial Matrix, featuring Dr. Eilís Dowd, Professor of Pharmacology and Therapeutics at the University of Galway. Today, she'll be highlighting her lab's recent data demonstrating that dopaminergic cell replacement in the Parkinsonian rodent brain, using both fetal and induced stem cell-derived cells, is enhanced when the cells are transplanted in a neurotrophin-enriched collagen hydrogel. Before we get started, we'd just like to acknowledge our partners for this series: the American Physiological Society, the Society for Neuroscience, the Federation of European Neuroscience Societies, the American Autonomic Society, and Scientist.com. And specifically, we'd like to give our thanks to today's host society, the Federation of European Neuroscience Societies, for partnering with us on Dr. Dowd's event. We'd also like to say a huge thank you and a shout out to all of our series sponsors. None of this would be possible without them. And now, without any further delay, I'm going to welcome Eilís. Hello, good afternoon.
Hi, Sydney. Hi. Thank you very much for having me here today. I'm really excited to give this talk. Can you see my screen now?
Yes.
Sydney? Super. Okay.
Look forward to it.
So, yeah, as I said, I'm just really happy and really excited to give the talk here today. I'd love to know where people are from. I was just in the chat. Actually, do you know what? I'm going to come out of my talk for a second, Sydney, if that's okay. And just see where people, oh, there's a few people from, wow, okay, India, New Jersey. Okay, yeah, just I'd love to know as well why you're on this talk today. Are you medics, clinicians, scientists, students? Just be great to have an idea of who I'm speaking to when I'm giving this talk. So you can pop all that in the chat. It'll just give me a bit of context for what I'll be talking about. Okay, so I think I'll just start. You should see the screen now in presentation mode. So I just thought, yeah, again, just really want to thank Professor Dennis Brown and Patricia in the American Physiological Society for the invitation today, Sydney, who you met there, for looking after me so well, and also my colleagues in the Federation of European Neuroscience Societies who suggested me for this talk today. So thanks to everybody in FENS, and also to SFN and the American Autonomic Society as well. I thought I'd just start by giving a little overview of myself, just to give you a feel for where I'm coming from. So I am here in a city called Galway on the west coast of Ireland. So if you draw a line straight across from Ireland, you will hit Newfoundland in Canada. So I'm pretty much right across from here, straight across the Atlantic Ocean. So I've been here in Galway since 2005, but I'm from Galway originally as well. So I'm really lucky to be back here working in my hometown. After I did my PhD at the University of Edinburgh up in Scotland, and after that I spent some time in Cambridge as a postdoc, and then I went across to Montreal to McGill for a couple of years on a Wellcome Trust fellowship, postdoctoral fellowship, and then came back to Cardiff University. So I spent about 12 years outside of Ireland altogether. But then I was really lucky to get the job back home in Ireland. And it's beautiful here. Now it's cold and it's grey today, but I think it's cold and grey in a few places today. So just again, I've had a few leadership roles in neuroscience. So I've been president of Neuroscience Ireland, which is Ireland's national neuroscience society. It's a member society of the Federation of European Neuroscience Societies. I was president of NECTAR for 4 years, and NECTAR is the Network for European Central Nervous System Transplantation and Repair. So it's a society in Europe that oversees all the work into central nervous system cell transplantation and restorative therapies for neurodegenerative conditions like Parkinson's and Huntington's and Alzheimer's. I've sat on the governing council and the executive committee of chaired FENS, and I chaired the FENS communications committee, and I have several editorial roles as well. The work I'm going to talk to you today is around brain repair for Parkinson's disease. So the repair and construction of the Parkinsonian brain that can be, theoretically so far, a little bit practically as well, but I'll take you through that from cell-based therapies. And we made a documentary about that a few years ago, and I have a couple of clips of that documentary in the presentation that I'm going to show you just now. And Sydney has also uploaded it in the handouts, a link to it, so you can watch it if you like. It's only about 25 minutes long, but it kind of sets research in the context of the patient. So really it features 3 Irish patients with Parkinson's and what it's like for them living with Parkinson's, and then it kind of sets our research in that sort of clinical context. Okay, so let's get started. So Parkinson's means a lot of different things to a lot of different people. And if I spent the 45 minutes just talking about Parkinson's and what it means, I still wouldn't do it justice. So I'm not really going to talk a whole lot about Parkinson's disease. I'm going to let Tom Isaacs give you a little bit of a flavor of what the condition meant to him. So Tom was diagnosed with Parkinson's when he was 26, and he went on, because he was young and really healthy. Of course, he had Parkinson's, but he was really healthy and fit. He went on to fundraise and he established a charity in the UK called the Cure Parkinson's Trust, now called Cure Parkinson's. And he died age 49 in 2017. And he was a massive loss to the Parkinson's community globally. He was a hero in Parkinson's research and in Parkinson's advocacy. So just a little bit of a click. He's like the British Michael J. Fox. A little bit of a click to hear what Tom had to say. So Tom came to speak at a conference I was organising on brain repair in Galway in 2014. And this is some of the material he sent me for that conference.
When you're diagnosed with Parkinson's, life changes beyond all recognition. Socially, it is a very visible condition, yet to society it seems to be invisible. No one ever talks about it. It is, to many, a private misery. No one ever mentions becoming frozen overnight in one position and the ongoing sleeplessness. No one ever talks about the side effects of the complex medication regime, which can be as extreme as the Parkinson's symptoms themselves. And no one ever talks about being imprisoned in your own body. And no one ever talks about the psychological changes that face people who live with this chronic condition. Parkinson's removes control. It destroys natural movement. It can be painful. Parkinson's challenges everything in life that you used to take for granted.
Okay, so wonderful, really, really powerful. Anytime I heard Tom Isaacs speak, it's just impossible not to be moved by him. So just, we all know Parkinson's. Probably some of you may know Parkinson's a lot more than I do. Maybe some of you live with Parkinson's, but Parkinson's is primarily, it was originally recognized as a movement disorder. But of course, there are a lot of non-movement related symptoms associated with Parkinson's as well. But in the context of brain repair, the idea of brain repair, it's really largely to repair the movement impairment associated with Parkinson's disease. So just to take you through some of the histology or some of the pathophysiology that's related to that. So Parkinson's was first described in 1817, and it took about 100 years later before pathologists recognized that these pigmented cells in the ventral midbrain were missing from the brains of people with Parkinson's disease. And several decades later, before the neurotransmitter dopamine itself was discovered, but very soon after the discovery of dopamine, it was found or reported that this neurotransmitter is deficient in people with Parkinson's disease. So the link was really quickly made between the loss of these cells, this new neurotransmitter that was discovered, and the dopaminergic impairments in Parkinson's disease. And then within a very, very, very short time, this drug, levodopa, was first administered to patients. So 1961, which is nearly 75 years ago, 65 years ago. I can't do the math. I can't do maths when I'm giving a seminar. But this drug was first administered to patients, and it was extraordinary. So this is the original report of the administration of levodopa to people with Parkinson's. So the effect of a single intravenous administration of levodopa was, in short, a complete abolition or substantial relief of akinesia. Bedridden patients who were unable to sit up, patients who could not stand up from a sitting position, and patients who, when standing, could not start walking, performed all of these activities after levodopa with ease. They walked around with normal associated movement, and they could even run and jump. So this was an absolute miracle drug. And if you ever saw that Oliver Sacks movie Awakenings, it was levodopa that was being administered to patients in that film, which is a film based on real incidents with that neurologist, Oliver Sacks. So absolutely amazing wonder drug. But here we are, what are we, 40, 50, 65 years later, and this drug is still the very best drug that we have for treating Parkinson's disease. So I find that amazing. Look at all the decades that have elapsed between 1961 and today, and this is still the best drug that we have. So this drug works because it's the biological precursor for dopamine, the neurotransmitter. And when people are administered levodopa, it gets enzymatically converted into dopamine in the brain, and it serves to replace dopaminergic function. So I've another little clip. Oh yeah, and just to give you a little bit of context for where I'm going to come back to in a little while, about 20 years later, the concept of brain repair for Parkinson's was born, about 20 years after the first administration of levodopa to patients. So I have another little clip just about dopamine and the role that it plays in movement. This film was made for a lay audience, so it may or may not be suited to the audience that I'm speaking to today, but it's only about a minute long. So
how we move, how we think, how we feel, how happy we are, how sad we are, all of those things are controlled by the chemical signals in our brain. Dopamine is produced from very early on in development. So within about 6 weeks after the egg is fertilized, the dopaminergic neurons are born. They're born as little round cells, and then they gradually grow out into the front of the brain where they release dopamine. It's the front of the brain then that regulates the movement. There's a whole circuit of different neurons releasing different messengers that are required to become engaged when somebody wants to move voluntarily. And dopamine acts a little bit like a lubricating fluid. In Parkinson's disease, it's the brain cells that make dopamine. Some of those brain cells start to die and degenerate. So we have less dopamine in the brain. So because we have less dopamine, patients with Parkinson's disease are not able to move as well. So they lose control of their movement, lose control of their motor function. By the time they start to develop symptoms, about half of their dopamine neurons can already have died. And they could have lost up to 80% of the dopamine in this part of the brain where dopamine is required.
Okay, so that's a little clip from Feats of Modest Valor, and I just want to just say that Feats of Modest Valor has nothing to do with our scientific endeavors. The Feats of Modest Valor refers to a beautiful poem by Robin Morgan, an American poet who was diagnosed with Parkinson's, and I've uploaded that poem in the handouts as well. It's absolutely beautiful, and it's about the feats of modest valor that people with Parkinson's face every day in trying to do their ordinary everyday tasks. So it's really beautiful. If you're interested in Parkinson's or poetry or piece of art, just go and have a look at that poem. It's stunning. Okay, so the concept of brain repair then it's really a very simple concept. If those brain cells that are regulating movement are dying, and it's the loss of the brain cells that are contributing to the major symptoms of Parkinson's, then the idea is simply it's a transplantation study. Can we transplant healthy cells back into the brain? And that is a concept. And I said that concept first started being tested, about 20 years after levodopa was first administered to patients. Now, where do we get healthy cells from? And for many decades, there was only one place. Until stem cell technology, came to the fore, there was only one source of healthy cells. And that was, a brain cell transplant. We could harvest the dopamine neurons and transplant them in healthy dopamine neurons and transplant them into patients with Parkinson's disease. But you saw in that little clip that, you know, the cells, the cell bodies are in the back of the brain, and they send out axons to the front of the brain, and they branch a lot up there. And you can't take them from an adult, like cadaver, like you would take a kidney or a heart for transplantation, because you try to isolate those cells, you'll sever the axons and the cells will die. So the only place they could be taken from is when there are little round cells shortly after birth during normal development. So in countries where abortion was legal, scientists and neurologists could use those, the fetal tissue to harvest dopaminergic cells for transplantation. And over the decades, there's been about 100 patients that have been transplanted with fetal cells. So that was the first source. And we have a really incredibly new source. I mean, we've been hearing about stem cells for a long time, but actually the very first stem cell trials for Parkinson's in this kind of stem cell trial only started 4 years ago. So it's really new. So I'm sure you all know, but just in case, just to make sure we're all on the same page. So a stem cell is a cell that can replicate itself and differentiate into different cell types. And the ones that are the types of stem cells that are being investigated for brain repair in Parkinson's are embryonic stem cells and induced pluripotent stem cells. So just one other slide, just to give a bit of background. So the embryonic stem cells are the ones that are taken from the inner cell mass, and they form embryonic stem cells, and then those stem cells can be directed to differentiate into whatever type of cell is required for regenerative medicine. So in the context of Parkinson's, we want to convert these embryonic stem cells into dopamine-producing dopaminergic neurons. But the cell type that we're interested in, we're not actually permitted to work with human fetal cells in Ireland or with human embryonic stem cells in Ireland for legal reasons. We can work with induced stem cells. So I'm sure you've all heard of this concept, but induced stem cells is where, you know, after a skin biopsy, the dermal fibroblasts can be plated down and then reprogrammed, engineered back into a stem cell state. And those are called induced pluripotent stem cells. And then they can be differentiated on into dopaminergic neurons. So I just want to give you a quick timeline of this sort of overview of brain repair. So as I said, the very first transplantation of fetal cells, so this was fetal rat cells into rats, so this was a model system, was 1979. And about 10 years later, the very first patients were transplanted. So that's a long time ago now, 1987, even though this sounds, to me, I always think this sounds like something that must be really new, cell transplantation, brain repair. But 1987 was actually the first transplantation in Sweden. And I view myself as a granddaughter, a scientific granddaughter of this era, because I did my postdoc with a professor called Steve Dunnett. And Steve Dunnett did his postdoc with Anders Björklund, which was one of the team of Swedish investigators that were working on this from the very, very beginning. So in the intervening decades, then there's been several clinical trials of fetal human cells in Parkinson's patients. As I said, about 100 patients have been transplanted. The cells are transplanted directly into the brain, and they replace the cells that die in Parkinson's disease. And then more recently, the stem cell era has begun, but I'm going to come back to that in a little while. So this era was really important. This is the era that, you know, gave rise to the whole concept of brain repair for Parkinson's disease. And these 100 patients have shown that human fetal cells transplanted into the human Parkinsonian brain, number one, can survive, which is amazing. When you think about what those fetal cells, where they're coming from, the nourishing environment they're coming from in the developing brain, into the older diseased adult Parkinsonian brain, but they can survive, and they survive for decades. They grow, and they grow out axons, and they reinnervate the tissue that has been depleted of dopamine and dopamine neurons. They can release and replace dopamine and restore movement to the patients, and they can actually reduce medication needs. So there's a lot of collective data from those patients that have been transplanted to show this. And this is just one example. This is a case study of patient 7, published about 10 years ago. And you can see that this is the motor score. So the higher they are here, the worse their motor symptoms are. And then over 5, 10, 15, nearly 20 years that the patient was followed up. And you can see that after their first transplant, the motor scores very quickly started to come down. And then with the second transplant, they came right down. And between, for the last, within 5 years after transplantation, they no longer needed dopaminergic medication. And the dopaminergic medication that they would have been on would have been this drug, levodopa, the wonder drug. Now, as I said, levodopa is a wonder drug, and you saw that first description of the use of levodopa in patients, but it is really problematic because within a few years of taking levodopa, people start to develop very severe side effects. And you saw Tom Isaacs there in the original film, and he wasn't able to control his movement properly, and he had a lot of abnormal movements. That was not his Parkinson's symptoms. That was actually the side effects of the drug. So people that take this drug, levodopa, this wonder drug, for a period of time start to develop dyskinesias, abnormal movements. So it is a wonder drug. Absolutely. That's one message I really want to get across. We're really, people with Parkinson's, at least there are fantastic drugs for this condition. But of course, they do eventually lead to some troubling side effects. Okay, so the recent stem cell trials then that have just started are trying to basically emulate what happened with the fetal human cells over those decades, but using a more ethically acceptable and logistically workable source of cells. So one of the major issues with this era, with the fetal transplantation, you might say it's worked so well. Look at that patient 7. Why aren't all people getting Parkinson's brain repair? Why aren't we getting fetal transplants? Where there is fetal tissue available, why isn't it being used for transplantation? And there was one major problem that's really held it back from proper clinical translation. And that apart from the ethical issues of using human fetal transplants, one fetus was not enough for humans, for patients, because very few of the cells that were transplanted actually did survive. I said they survived, yes, they did, but very few of them. So about 95% of the cells would die after transplantation into the brain. So in order to get enough dopamine cells to restore and repair the Parkinson's brain, we needed up to 12 human fetuses for every patient. This is a massive issue. This issue of poor transplant survival really exacerbated this ethical issue of the human tissue and the logistical issues of having all of that tissue available for stereotaxic transplantation into a patient in hospital. So we started to think about this. Why are and we were not the only ones. For many years, people all over the world in this field were trying to tackle the issue of survival. Why are they not surviving? And there are several different reasons. So one of them is that they die by a form of apoptosis called anoikis. I love this word. It's a Greek word. And if there's any Greeks listening to me, they will know it means to be without a home or to be rendered homeless. So these cells are dying by a form of apoptosis after they are made homeless. So the cells are taken out of their tissue. They're dissociated from their tissue. They lose their anchorage to the extracellular membrane, and they die by this form of apoptosis. That's one reason the cells are not dying. One reason cells are not surviving. The other reason they're not surviving is, you have to imagine an embryo or a fetus developing in utero. It's getting all of the neurotrophic factors, the growth factors, the neurotrophins that are required for those cells to develop into dopaminergic neurons. And we're harvesting that tissue, dissociating it into single cells, and then transplanting it into the brain of a patient. So they undergo growth factor deprivation, and they die because they're not getting the signals that they need to survive and grow into neurons. And then one of the other really important reasons, of course, is you're transplanting one person's cells into another person's brain. So you can suppress the immune system, and of course that was required with immunosuppressive drugs, but there's still a massive inflammatory response in the brain, and that attacks and damages the cells that are transplanted. So we were, around this time, as I said, I'd been away for 12 years, and I came back to Ireland in 2005, and there was what was to become the largest ever science foundation in Ireland. So Irish government funded research centre in Ireland was just kicking off in my university at the time. And it was a research centre on material science and biomaterials in particular. So we started to team up with some of the material scientists in that research center and to think about, could we transplant our cells in a material that would, we kind of thought if you can gel or encapsulate the cells here in pink in a material before you transplant them into the brain, that the material could act as a microenvironment for the cells in the brain. And we could, for example, like if we used a material that the cells could attach to, like a scaffold, then that would reduce anoikis because you're giving them an extracellular matrix to attach to. We could also enrich this gel with neurotrophin, so the neurotrophic factors that the cells need. And the gel might also act as a shield against the host immune response. So these were my colleagues at the time, Abe and Ben, still collaborating very closely with them. And after a bit of trial and error, we decided on collagen. So collagen to be our material to make this gel. So, well, firstly, collagen does form a gel, but it's in situ gelling. What that means, it's thermoresponsive. So when it's outside of the body and cold, it's in a liquid form. So we can inject it through stereotaxic techniques directly into the brain in a really minimally invasive way. So it wouldn't cause any extra damage to the brain. We don't have to form gels and then cut a hole in the skull and implant a gel. This is a really injectable gel. It's natural, biocompatible, biodegradable. It has this amino acid sequence for cell adhesion. So it forms an artificial extracellular matrix for the cells after transplantation. So we had this concept, that's all very well and good. We can all have good concepts. So we set about, of course, testing it. So this is about 10 years of work I'm going to whiz through now. So there's just a little bit more. This is another diagram from a review we published recently. So just to have it clear, we have our cells, and they are going to be encapsulated within a collagen that's cross-linked. The cross-linker just serves to gel the collagen, and we're going to also enrich that hydrogel with neurotrophic factors. So depending on how much cross-linker we add into the collagen, we can make it a stronger or a weaker gel, so more or less gelated. And as I said, that then the idea is that would provide structural support, an enriched microenvironment because of the neurotrophin enrichment, and a physical barrier against the immune cells. Oh yeah, I have another little clip from Feats of Modest Valor, if that's okay. There we go.
Because cells die in Parkinson's disease, we're going to transplant stem cells into the brain. In this context, we're going to be harnessing biomaterials to encapsulate the cells.
What we have here is collagen. So this is the natural biomaterial which we are using, and we use it because it's very biocompatible, very suitable for implantation into the patients that we are aiming at.
So we can make the gel at a consistency that matches the human brain.
Exactly.
At room temperature, it's like water. When it's injected into the brain and it hits the temperature of the brain, it forms into a gel. That gel then can be used as a scaffold or a physical matrix to support stem cells that are transplanted into the brain. Embedded within the hydrogel, we're also going to have tiny little microscopic spheres. What they're going to do is encapsulate anti-inflammatory factors and drugs that protect the neurons from dying. The drug that's showing the most promise is called GDNF, which is a chemical that our brain makes that helps neurons to grow. So what we can do is inject the GDNF into the adult brain where the neurons are dying, and to try and stop the neurons from dying at that point.
Okay. SEM. So you can see that it is a porous gel, so it's full of lots of little pores, And that is really nice because it forms a little niche that the cells can sit into when the gel when the collagen is actually gelled. And we can, as I said before, depending on how much cross-linker we add to the collagen, we can control the pore size. And over this time, we found that the particular amount of cross-linker that best suits the cells we're transplanting. And this shows a cell encapsulated hydrogel. So the cells are in here inside the hydrogel. Again, this is just ex vivo, just by SEM. Then, so that's the concept of having a niche for the cells and the cell adhesion and the sort of reduction of anoicus. We've done a lot of other things around that, but I just want to get onto the functionality really. But the next hypothesis we had was that the gel could retain the neurotrophins for longer in the brain. So we published a few papers where we showed that. So for example, this is GDNF, which is glial-derived neurotrophic factor, which is one of the primary neurotrophins for dopaminergic neurons. So if you inject a bolus of GDNF, just inject the protein directly into the brain, it's gone very rapidly within a day or two. But if you inject it within the hydrogel, which is gelled in here, you get a lot of retention of GDNF in the striatum for longer periods of time. Again, we can play around with the cross-linking here to control the retention of proteins. This is interleukin-10, which is an anti-inflammatory cytokine, which we also did some work with. So yeah, so the gel can provide a cell niche for adhesion, and it can retain proteins in the brain after transplantation for a short period of time. I'm going to get back to that. As you can see here, by day 4, the GDNF itself is gone, and actually the hydrogel is gone as well after a few days, but that can be beneficial. Okay, then we also wanted to know if the hydrogel could actually shield the transplanted cells from the host immune response, and it did that in a really, really significant way. So these are some cells that are transplanted into the brain here on their own with neurotrophins, and then in the hydrogel on its own, or the hydrogel with neurotrophins. And this is a microglial staining. So you can see that when the cells are put into the brain without the hydrogel, there's a massive microglial or innate neuroinflammatory response to the cells in the brain. And these are astrocytes, and you get the same thing, a lot of innate immune responding to the transplanted cells in the brain. But if the cells are put into a hydrogel, the microglia and the astrocytes just don't respond to the cells. So the hydrogel is actually able to protect the transplant from the innate immune response. And that's it quantified, and we showed this again by a lot of different ways. This is another paper where you can see here again, this is the transplant, and here in the striatum, and there's a huge amount of astrocyte staining around the transplant when the cells are transplanted on their own, but very little when the cells are transplanted in the hydrogel, and that's it quantified there. And that's it visually. So this was a GFP expressing transplant, and here are the astrocytes around the transplant. Okay, so that is okay. So the hydrogel can, as I said, form a niche, have a niche for the cells. It can retain neurotrophins around the cells, and it can shield the cells from the host immune response. But does that actually then improve brain repair, which is what we are looking at? So I'm a preclinical researcher, not a clinical researcher. So this is how we do our studies. So we use Parkinsonian rats, and we test them for behavioral ability, motor ability, and then we can do a reparative cell transplant. So implant our cells, our stem cells, or our fetal cells. I'll get onto that in a second. Test again to see if motor function has improved, and then have a look via primarily histological methods if the brain has been repaired. So this is our very first study on the hydrogel in terms of brain repair in Parkinsonian rats. And it was published about 5 or 6 years ago now at this stage. So what you can see here is that these are fetal cells, but they're rat fetal cells. So this is a model system. We're transplanting rat fetal cells into the rat brain, the rat Parkinsonian brain. So what you can see here is this is the transplant here. When the cells are put on, I know you can't see it that clearly, when the cells are put in on their own, and then this is the transplant when they're put in the neurotrophin-enriched hydrogel. And you get a lot more cells surviving, and you also get a lot more re-innervation of the surrounding tissue that has been denervated by the Parkinson's lesion. So looking at it quantitatively, this is the number of cells that are surviving on their own. This is and then when they're implanted in the neurotrophin-enriched hydrogel, we get about 5 times more cells surviving, which was really dramatic, and about 3 times more reinnervation of the striatum. So that was really exciting for us. And of course, we had to look at motor function as well. So this is a rotational test, and it's because our animals, when we do a lesion, we do it in Parkinson studies, we mostly do it just on one side of the brain. So there's a motor dominance on one side where there still is a dopaminergic innervation, and that drives the animals to turn in circles, and that's really easily quantifiable. So everybody that does Parkinson's work with Parkinson's model looks at this rotation system. So when they're lesioned, they're all turning. And then after transplantation, they're all improving because all of the animals are getting cells. So this is ventral mesencephalon, so they're all getting fetal cells. But over time, the group that improves the most is the ones that get the fetal cells in the neurotrophin-enriched hydrogel. So this shows that not only were we getting better numbers of cells surviving, not only were we getting better reinnervation of the striatum, we were also getting improved motor function. We followed this up with another study, which we published in the FENS journal, the European Journal of Neuroscience. So this time we used, for different reasons, we used a younger donor age of the fetal tissue, again, rat to rat. So you can see here, again, that this is what the transplants look like when they're put in on their own, but when they're put in with the neurotrophin-enriched hydrogel, they're much better. They're much better able to reinnervate the brain. So looking at this quantitatively, you can see that there's a really big increase in the number of cells that survive when the cells are transplanted in the neurotrophin-enriched hydrogel. And the hydrogel itself actually is beneficial in this study. And when we look at behavior, again, they all come down because they've all been given a reparative cell transplant. But the group where the behavior improves the most is the ones where the cells are transplanted in the neurotrophin-enriched hydrogel and the cells that are transplanted in the hydrogel alone. So again, that was all really exciting. It got a lot of press, which was really exciting for us. Again, on the west coast of Ireland, in a university here, to get this kind of press was fantastic. So the British Neuroscience Association described it as a groundbreaking study by neuroscientists in Ireland, which is always nice when the British say that about the Irish. BBC Science Focus magazine picked it up and called it one of 5 incredible advances in brain disease treatment, which of course it isn't. It's conceptual and it's in a model system, but you know, we're not responsible for what the journalists say. But it got a lot of press, and that was fantastic because it led to us getting a grant for the Michael J. Fox Foundation to go to our next phase. But I'll get back to that in a second. So the next phase is, of course, that right now we are no longer in this fetal era. So we've made a hydrogel. It works brilliantly for rat fetal cells transplanted into a rat model. But where does that leave us for the stem cells? Because that is where this field is moving to now. So there's 3 stem cell trials that are running at the moment. The first one is using induced stem cells, and that's in Japan. Blue Rock Therapeutics, Lorenz Studer, is running one in the US. It's an embryonic stem cell trial, and the very first embryonic stem cell trial in Europe was just approved in 2022, just last year. So the question is, of course, can our hydrogel improve the outcome of stem cell-derived brain repair? Now, conceptually, these are really different cells, so they have different challenges to the cells that are being taken from the human fetal donor, but they still have their own challenges, and this is one of the major challenges. So this is a protocol where you take the stem cells, whether they're embryonic or induced, and you bring them through different stages to maturity. So at this point in time, at day 16, you can leave the cells in the cell culture dish and you bathe them with the neurotrophins that they need. You bathe them with GDNF and you bathe them with BDNF, and you give them all the signals that they need to become dopamine neurons. And they mature fantastically. Within about a month, they turn into mature dopaminergic neurons in the cell culture dish. But if you want to transplant them, if you want to actually take them for transplantation, you have to take them at day 16, because if they start to grow out these axons and processes, and then you lift them from the cell culture plate, you will sever them, you will kill them, and they won't survive after transplantation. So you have to take them at this stage when they're progenitors, when they're still round little cells and they haven't started putting out any outgrowths yet. But actually, when you take the stem cells at that point in time and transplant them into the brain, the survival is really poor, the differentiation is really poor, and it can take up to 20 weeks in a model system for any maturation to occur. So this is one of my PhD students went through all the literature, the preclinical literature, looking at around 30 different papers where they did cell transplant in model systems. Now, remember, there's no published data of stem cell transplants in Parkinsonian humans yet. So these are all preclinical studies. And you can see that the median differentiation of the cells that are transplanted is less than 3%. So, you know, even though these trials have started, and there's 3 trials running in Asia, the US, and Europe, the actual number of cells that are transplanted that will convert into the dopamine neurons that are required the percentage is only around 3%. So really something needs to be done to help these cells to survive. So, you know, along comes our concept, which is the hydrogel, which is if we can encapsulate the cells in the hydrogel that's full of the neurotrophins that they need, and this neurotrophins can bathe the cells for a period of time after transplantation, will that help their survival and maturation? So that is the question that our very first Michael J. Fox grant, which was awarded 4 years ago, that's what they wanted us to address in that question, to see if our hydrogel could improve iPS-derived brain repair. And this is some of our data. Okay, so the usual approach, with the cells on their own, cells in the hydrogel, cells with the neurotrophins, or cells with the neurotrophin-enriched hydrogel. And this time we're using human iPSC-derived progenitors. So we're transplanting dopaminergic progenitors taken from human skin fibroblasts-derived induced pluripotent stem cells. And this is some of our, I'm going to skip through that first study because I don't really have time for that. Okay, so one of the studies we did was because we are doing now human cells into rat, we have to avoid transplant rejection. So one of the ways we could do that is to use nude rats as our host rats. They are rats that don't have a thymus, so they don't make functional T cells. So we avoid having to immunosuppress the animals for long periods of time, which is really good from an ethical perspective. So what happened with our cells? So these are human nuclei. So where you can see the brown dots here, that's the transplant. So this is the number of human nuclei that are in the rat brain. So the survival of the transplant when they're on their own versus in our hydrogel. So you can see already there's a lot more human cells surviving in the rat brain when they're in the neurotrophin-enriched hydrogel. And when we quantify that, actually between the cells on their own and the cells in the neurotrophin-enriched hydrogel, these two groups, there's an 8-fold improvement in graft survival in terms of actual number of cells that survive the transplantation process. This is another stain. This is a cytoplasmic stain. So it's a human cytoplasmic stain. So again, you can see the grafts when the cells are put on their own, which is what's happening in the 3 trials that are running at the moment. The grafts are really small, but when they are transplanted in the neurotrophin-enriched hydrogel, we get much bigger grafts. In fact, we get 13 times bigger grafts when they're transplanted in the neurotrophin-enriched hydrogel. Okay, so that's survival, but all we're talking about here is human nuclei and human cytoplasm. What about the actual dopaminergic differentiation, which is what is key? That's one of the things that's really low in all of the preclinical studies. As I said, a median of less than 3% of the cells survive. So this is, I'm going to go straight onto this. So any of these brown cells here, there are dopaminergic neurons, and you can see that there are a lot more dopaminergic neurons when they're transplanted in the hydrogel. And actually, this is the volume. You can see that the size of the graft is much bigger, but when we actually count the cells, there are 16 times more differentiation of human-induced pluripotent stem cells into dopaminergic neurons in the rat brain when they're transplanted in the hydrogel. So basically, this hydrogel is having a really pronounced effect on the survival of and differentiation of human-induced stem cells in the Parkinsonian rat, which is fantastic. We did some correlation analysis. You can see that, but I'm not going to go into that. Okay, now our last study, the nude rats are brilliant. They're a fantastic model, and they're actually really clinically relevant as well in terms of autologous transplantation when you use the patient's own cells, harvest them, turn them into dopamine neurons and transplant them back in, because there's going to be no T-cell mediated response. But actually the leaders in this field at the clinical side say that really what we're going to be doing is banking having stem cell banks, and people will, if this becomes a therapy in the future, people will receive allogeneic transplantation from HLA matched cells, and they will undergo immunosuppression. So we wanted to know if this would work in immunosuppressed animals, not just in nude animals. And actually what we found was that it didn't work. So these are the nuclei. This is the number of cells surviving, again, human cells in the rat brain. And when we look at survival, there's no beneficial effect of the hydrogel. And we look at maturation, there's no beneficial effect either when the cells on maturation or dopaminergic differentiation in immunosuppressed animals. So that was really puzzling. However, we did look back at our protocol, and we just immunosuppressed for a day before we did transplantation, which is typical, but we found a lot of T cells at the graft site. So a lot of CD4 positive and CD8 positive T cells infiltrating into the brain. So basically what we think is that our immunosuppression protocol was not sufficient to immunosuppress the animals before we did the transplant. So even though the cell survived, the hydrogel wasn't able to protect against an adaptive immune attack. But very happy to say that Michael J. Fox have just funded a continuation of this research. So the new grant just started in September, and they want us to they want us to get this hydrogel working in immunosuppressed animals, because that has the most clinical translatability. So that's what we're doing at the moment. So this grant just started, but we've got some really interesting pilot data already where we did a proper immunosuppression, and we can see just with our little pilot that there's the hydrogel is starting to emerge as being beneficial even in immunosuppressed rats. But that study is just started. Okay, so that's it. I just want to thank everybody, all my PhD students that did this work over the years, my collaborators in Galway, and also Thilo and Nicola in Edinburgh. Funders and patients that Tom Isaacs that you saw, Tom, Elaine and Brian that feature in Feats of Modest Valor, some members of early onset Parkinson's here in Ireland that I work with closely. And I want to leave you with one final thought, and that is this. Okay, this is one of the most amazing images, and I use this image all the time in my teaching. And this is an image taken from the Journal of Neuroscience in 2019 from an article by Matsuda et al. And this image never fails to astonish me. This is the terminal branching of one single dopaminergic neuron in the mouse brain. So that is one neuron you're looking at. When you look in a textbook and you see a neuron and you see a cell body axon and the terminals as a little Y-shaped thing, this is the Y part. This is the terminals. So our brains are made up of 100 billion of those, plus another 100 billion glia, and 1,000 trillion connections between our 100 billion neurons before we even start talking about the connections between the glia and the neurons. So the complexity is just unreal. The complexity of what's in there and the damage that we're trying to repair with cells. Okay, and I think that's it. Will I come out of that, Sydney?
Yeah, thank you so much. Really, really fantastic presentation. Really powerful, and those videos were just amazing to hear from those people. And I definitely will be watching that movie.
Thanks, Sydney. Thanks. Does anybody have any questions, or would anybody like to ask me anything? Or
yeah, we do have some questions already, and hopefully we'll get lots more. Just before we do jump into the Q&A, okay. I would just like to ask everybody, if you do have to leave early, please send in your questions, thoughts, comments into our survey as well, and let us know what you thought of the webinar today. And then, yeah, that's it. So let's jump into the Q&A. So our first question: how many cells are implanted, and at what time can one observe an increase in the dopamine levels before seeing an improvement in the motor function?
Oh yeah, that's a great question. So in our model studies, we transplant somewhere hundreds of thousands of cells. So you know, with the fetal cells, we would have transplanted about 1 million, but only a percentage of those are dopaminergic because you're taking them from fetal tissue. So there's not just dopamine neurons in there, there's glia and there's other neuronal types and all of that. So we have to transplant a lot of cells to get the number of cells that we need for repair. And how long? Usually a long time. Again, in the IPS, in the stem cell derived brain repair studies, we need about 6 months before we start seeing anything. So these are really, really, really long-term studies.
Okay, thank you.
And you saw in the patient, I did have it off. It was about a year after transplantation when the improvement was first detected.
Okay, excellent. Thank you. Based on the figure you showed earlier, this was maybe halfway through your presentation. Hopefully the question will make it a little more clear. Are these being implanted into the striatum instead of the SNC?
Yeah, great question. Yeah, these are all ectopic transplants because dopamine is required in the striatum. So it was shown over many decades of preclinical research that the best place to put the transplants is to put the cells into the striatum. So you're not actually putting them back where the cell bodies are in the nigra, because there's quite a bit of distance between the nigra and the striatum. So yeah, they're ectopic. They're actually in the wrong place, but they still serve to connect to the host striatal cells, the medium spiny GABAergic neurons in the striatum, and form functional connections. And that has been shown to be the best place to put them. Okay,
excellent. And I think a good follow-up question would be, why are you using a lesion model for Parkinson's disease? Would it not be more interesting to use a viral synucleopathy model? That would allow for testing how transplanted cells get affected by synucleopathy and neuroinflammation.
That's somebody who knows what they're talking about there. Yeah, it's a brilliant question. And I suppose most of the brain repair studies are done in lesion models. So the idea is you can inject a neurotoxin that selectively kills dopamine neurons, and that'll wipe out the dopamine neurons on one side of the brain. So we have a really clean, albeit artificial, lesion where you can just, there's no dopamine neuron. So anything you put back in is transplant derived. And that's been the classic model for studying brain repair studies. But yes, I don't know if anybody heard of the prion concept of Parkinson's, but one of the key pathological, I'm going to start talking now for a couple of minutes. Is that okay, Sydney?
Absolutely, we've got time.
It's a great question. So one of the key proteins involved in Parkinson's pathology is called alpha-synuclein. And it was from 2 transplanted patients, human patients that were transplanted with fetal cells decades ago. They died for other reasons, unrelated reasons, but their brains were donated to science, and the neurologists could section their brains and have a look. And they found that even though these were given fetal cells, the fetal cells had Parkinson's disease. They had alpha-synuclein pathology. That was 2 papers published back to back in Nature in about 2006. I think so. That was the first time that showed that somehow the alpha-synuclein pathology was transferring into the transplanted cells. So that is what this participant, this audience member, is asking about today. Why don't we study it in synucleinopathy models so we can see if the synuclein transfers into the cells? And yes, we will. We absolutely will in the future. But first, we need proof of principle in the lesion model. In fact, it's one of my previous PhD students, Deirdre Hoban. She's lead author on a paper, the very first paper that did show that, and she worked as a postdoc for a while with the Swedish group with Malin Palmer and Anders Björklund, and she did show that stem cells, embryonic stem cells, do also gain this alpha-synuclein pathology when they are transplanted into a synucleinopathy model. So it will be critical to do that at some point. And actually, I'm really interested to see if the hydrogel can affect the transfer of synuclein in any way.
Okay.
Yeah, that was a long answer. That was a long answer.
I think it was great. Very detailed. Thank you. On that note, did you see any limitations using hydrogels?
I think no. I mean, they're biocompatible, they're biodegradable, they're already clinically approved. I think the only the only negative, if you want, is technical, because they do gel. So just technically, they can gel in the cannula sometimes when you're doing a transplantation. So cannula can get clogged and things like that. But that can be overcome with keeping the cannula a little bit colder and things like that. But we had a few lost cell preparations because the hydrogel gelled before it got into the brain. But apart from that, absolutely no downside.
That's excellent. Okay, thank you. Okay, next one, pretty cool. Agree. My guess is that a specific brain zone is needed to inject the encapsulated neurons. Is this correct?
Yeah, so that goes back to whether we're putting it into the substantia nigra or putting it into the striatum. So putting it into where the cell bodies normally reside, or putting it into the striatum where the dopamine is needed. And we target it into the striatum, or the caudate putamen in humans.
Perfect. Okay. Is there an immune response against the collagen protein in the gel?
Absolutely none, and which is fantastic. And in fact, you saw that it reduces the immune response of the host to the cells. And when it's put in on its own, there's no immune response at all. So it's not immunogenic.
Fantastic. Okay. Did you perform any further motor or behavioral tests besides the rotation test? And does the hydrogel have any further effect on the transplanted animal?
Yeah, that's a really good question. I think that, like, I come from I come from a lab. I was trained with Steve Dunnett in Cambridge and Cardiff, and he's really heavy into behavior, like really detailed motor analyses, because there's no point in having cells that survive in the brain and grow very well if they don't restore function. They're not going to be any good to patients. So we do a lot of different motor analyses in our different Parkinson studies. So we look at stepping tests, whisker tests, forelimb akinesia. We look at sensory motor neglect. There's a lot of different elements of function that we look at. But with these stem cell studies, we're really just focusing on the pathology at this stage as our primary outcome, just to see what's happening. We actually didn't, I didn't mention this, but we really didn't expect the hydrogel to have such a beneficial effect on the stem cell derived transplant, because it's a little bit complicated, but because I don't know if you noticed in the earlier slides that I showed that the hydrogel only retains neurotrophins for a few days in the brain. And we know that that's all that's required for fetal cells. They just need to get over the window of transplantation. But for the stem cells, we kind of thought that they would need to be bathed in neurotrophins for about a month. So we weren't expecting such a dramatic response. So if we had known there was going to be such a dramatic response, we would have done lots of behavior. We were just kind of going in for a quick. You saw the N was only 6. It was a relatively small study, but the effect was just phenomenal. So we do need to go back, do it again, and do full-blown functionality assessments.
Okay, yeah. Amazing, great. I think we have time for one more question. Just for everybody's knowledge, you are more than welcome to stick around to the survey and submit any more questions you have in there, and Eilís will be able to answer. So our last question, are regenerated astrocytes necessary for neuron cell repair?
Yeah, that's a really good question. And that's something that people have really overlooked in this field. So when we were using fetal transplants, you were taking a chunk of the fetal tissue and dissociating it and transplanting everything that was in there. So there was no purification of dopaminergic neurons. So we were transplanting dopamine cells, serotonin cells, GABAergic neurons, and lots of glia, lots of astrocytes. But since we moved to stem cell derived brain repair, the aim is to get as pure a population of dopaminergic neurons as possible. And really, we don't know what the outcome is going to be in patients because these trials haven't happened yet. They're just ongoing at the moment. So it's something that a lot of people are looking at. They are looking at trying to compare dopamine cells on their own, stem cell derived dopamine cells on their own, or stem cell derived dopamine cells with different concentrations of astrocytes. It's a really key question, yeah, because they're support cells, they release GDNF, they release a lot of things that are required for the neurons to function properly. So my guess is we will find out that we do need astrocytes.
Okay, yeah, we will look forward to what you find out. Thank you so much, Eilís. This was a fantastic presentation. Feel free to have a read of the chat. Everybody is saying really nice things about your presentation, and I have to agree with this last comment. Very easy to follow and well set presentation. So thank
super.
you.
Thank you.
Yeah,
Thank
you so much.
Thanks everybody for coming today to listen to me wherever you are. Really appreciate it.
Okay, fantastic. In closing, we hope you enjoyed this Inside Scientific webinar. Another quick thank you to all of our partners for helping make this series possible, and I hope you all enjoy the rest of your day.